We're in Lab 6 Microscope Lab. So in this lab, you need to be familiar with the microscope, its basic structure, how to operate it, and each part of the name. So the microscope have all these names. You need to be familiar with it: ocular lens, objective, and these terminologies. So microscope is very important in biology because it's like a big magnifier. So you need to be familiar with the microscope. And you need to know what's the name of this, what's the name of that. Like in the lab practical, I'll give you a microscope. I'll ask you, what's this? It's called the ocular or ocular lens. And I'll ask you, what's this? It's called the objective lens or objective. Don't tell me it's lens. You have so many lenses in the microscope. And this part is called the arm, and this is the stage. So the microscope is a big, you can think it's a big magnifier. It just amplifies the signal, but you have more than one lens. Basically, you have these two, right? Ocular and objective. So instead of amplify the signal once, it amplifies the signal twice. So you take this one, multiply by this one. It will be the total magnification. So it's a very powerful magnifier. And this is called the coarse adjustment knob. So it moves. The sample, you put them here on the stage. And the light come from the sample, will go through the objective lens, go through the ocular lens, and go to your retina. So apparently you need to focus perfectly on the retina. So the adjustment knob, you have the coarse adjustment knob, you have a fine adjustment knob. They just move the stage. So eventually they move the stage and make the light perfectly focus on your retina. And what's the big difference between the coarse and the fine one? The coarse one moves the stage much quicker. So the fine adjustment knob, you need to turn the fine adjustment knob 50 times, equal to 1 time of the coarse adjustment knob. So it just moves it slower. And when do you use the fine adjustment knob? That's when your sample are very close to the objective. Like when you are in the high magnification, like 10x or 40x objective, you want to use the fine adjustment knob. You don't want to use the coarse one because you can accidentally bump the sample into the lens. So these are the basic structure. It tells you how to use the microscope, and when you are using those high magnification, you use the fine adjustment knob only. So I will ask you the different part of the microscope, okay, and they show you how to put the slides on. They have different kind of slides, transverse, cross sections. Well, we talk about them in the first lab, right? Different kind of cut. And let's look at one more thing: the total magnification. What's the total magnification of the microscope? So if I give you the ocular lens is 10x, multiply signal by 10 times, and I use the 4x objective, could you tell me what's the total magnification? And the answer would be 10 multiplies 4, 40 times. Yeah, and that's actually the smallest magnification. And if I give you the 10x ocular in the lab, all the ocular lenses are actually 10x, and 10x objective, and the total magnification would be 10 multiplies 10, that's 100. And if I give you the 10x ocular lens and 40 objective, and it turns out the total magnification would be 10 multiplies 40, that's 400. So that's how we calculate the total magnification. So this microscope can amplify the signal 100 times, sometimes thousands, depends on how you use these two lenses. And the bigger the objective, the bigger magnification, but your sample will be closer to the objective lens. That's why you need to use the fine adjustment knob when you are in the high magnification. Okay, that's pretty much it.